So the caption is, "Hey, what's this Drosophila melanogaster doing in my soup?" And the butterfly one, whoever had done the editing knew something about species names that I forgot to mention when I put species names up. So add this to your notes. And that the butterfly one, the species name was in italics. And this one, it's not, as the editor didn't realize. In English, species names are Latin and are considered to be foreign. If you're handwriting, you're supposed to underline them. If you're printing, they're supposed to be italicized. Now it's weird, the other levels are all Latin, but the rule for whether that is considered a foreign language, that's why you're italicizing when you print stuff. If somebody's speaking French, it's supposed to be in italics too, if you're writing in English. But the other levels sometimes get italicized and sometimes don't. But species names are always supposed to be italicized. So I did forget to put that down under binomial nomenclature, under the two words. So how do we know what to put in what group? How do we know who's related to whom and how closely are they related? Are they so similar that they belong in the same species? Are they pretty similar but not exactly similar enough to be in the same species so they're in the same genus? Are they in the same genus but a little bit less related and then need to be in the same family and order and so on and so forth? Is that relatedness is a tricky thing to figure out, and it's why there's always arguments about these things, about how you're determining the relatedness. What's supposed to be happening the same way we do with people. How closely related are you to me? Well, how far back up our family tree do we go before we hit a relative that you and I have in common? If it's just, our grandparents, then we're cousins. And then it gets to be second and third and fourth cousins. It's a whole thing. And you can't always tell from the names. My genetics are in the 23andMe database. I think there's 1,400 relatives that have rolled out. Not a single one of them has my last name. My last name's pretty rare. But most of them are fourth to fifth to sixth cousins. But it's a level of relatedness, and there are definitions for those things. And so we'll go back to the bats. To be in the same order, two species need to share an ancestor that is also in that order. So two bats need an ancestor that they share that's still a bat. And that's actually one of the big discussions, is that the microbats and the macrobats go down the family tree. Eventually, those branches connect in a common ancestral population. Would that be classified as a bat? If not, they belong in separate orders. And whatever makes them a bat, one would assume wings and flight, actually evolve separately on those two separate branches. That's why they would be separated. But of course, you can't go into the past. Bats fossilize horribly, so you don't necessarily have a good fossil record to go with. You look at their genetics and, there's a lot of arguments about that. If you go into the past, and there's still a little of this, then you look for connections based on what can be done, on functions. You can see this old level of classification by looking at Genesis in the Bible. Genesis is an explanation for where everything came from. And they had it broken down to certain things, certain groups of things were created on certain days. And how did they classify things? Well, the beasts of the air were all created on the same day, because to the ancients, things that could fly had to be related to each other. They had to be in the same group. they were analogous to each other. So they would have put butterflies and bats and bees and birds all into the same group based on function, based on analogy. And sometimes we still do that if that's all we've got to go on. But it's not a good comparison, because it turns out that bats and birds and butterflies are not built the same way. That's homology. Homology is architecture. How is something built? And that is a much more reliable comparator. Oops, backwards. You probably can't read the labels, so it probably doesn't matter that it's backwards. All of these vertebrates have certain bone structures that show you connections. Is that, in this case, it's talking about the two bones right in here, there, in a bat. Vestigial bone doesn't, people say, oh, it's vestigial, it doesn't have a purpose. Vestigial technically means it doesn't serve the purpose that it does ancestrally or typically in other animals with the same bone. This is the flying dinosaurs, flying lizards, pterosaurs, a bird. If you ever have chicken wings, you're familiar with these two bones. The horse, the plesiosaur, the big paddly guys with the long necks that lived during the dinosaur era. Homology. You can look at the structure. This is how they look at fossils and figure out what they're looking at. A good paleontologist might get a single bone and look at it and go, oh, I know what this is, and I know what sort of animal it belongs to. This is why they decided that dinosaurs were giant reptiles, that they could assign them to a group. I think if they existed today, we'd give them their own group. They're different enough from the reptiles that they probably would warrant their own group if they were alive today, and we could look at a lot more of them, but we're kind of stuck with structure. Going back to the bats. The question is, okay, we've got the microbats and the macrobats. Wings are a characteristic that sets the order. The shared ancestor, did it have wings? If the answer is yes, then the wings in both groups are an ancestral feature. It goes all the way back to the shared ancestor. If they don't, if wings evolve separately on the two different branches, then it's a derived feature. And just the fact that they both have wings doesn't mean that they're related to one another. As that wings are a derived feature in pretty much everything that flies, with the exception of ballooning spiders, or there's not a lot of ways to get up in the air and stay up in the air without wings. But in butterflies and birds and bats, it's a derived feature, is that their ancestors, their shared ancestors did not have wings. So it is, again, a way of determining connectedness. Here's another set of terms for that. You might want to leave yourself some room that I didn't, because I'm not going to fill in the definitions here, but you may want to leave yourself some room. So somebody says, well, according to my research, the macrobats and the microbats, their shared ancestor was not a bat. Now, ultimately, the order should be monophyletic. It should only have relatives that are in there, that are all shared, and their shared ancestors in there. And if somebody goes, no, their ancestor wasn't technically a bat, what they're saying is this group that should be monophyletic, you've got set up, and it's polyphyletic. This is a criticism. Is groups are not supposed to be two separate groups that shouldn't be in there together. Is if there are, two or more groups in there that shouldn't be in there, it's a polyphyletic group. Then you get weird situations like paraphyletic. Paraphyletic is kind of an acknowledgement that, okay, we've got two groups that should be in the same group, but we're not putting them there. And again, it all has to do with how OCD you are. Birds are direct descendants of dinosaurs. They have dinosaurs on their family tree. And because of that, because of what we've discovered in the last 30 years, birds and dinosaurs are a monophyletic group. Birds should be included in the dinosaurs, which are included in the reptiles, which suggests the birds should be included in the reptiles. Nobody really wants to do that. So we go, well, we're going to consider the birds a separate order, a separate class, actually. And people go, well, yeah, but they should be, okay, yeah, they're different enough. Even though they share the ancestor, they're different enough. We're going to still give them a different big group, and it's a paraphyletic group. And this happens a lot, especially in well-established systems where somebody comes in and goes, whoa, these are more related than we thought they were. And this historically separate group actually should be inserted into this other group. And people go, no, we don't want to do that. We'll just consider it a paraphyletic group. Here's where biology runs into reality. Part of this is multi-kingdoms. Originally, there were 3 kingdoms in nature. There were the animals, there were the plants, and there were the minerals. And before very long, the minerals got kind of split off into their own system. They have their own classification system. And the plants and animals as living things those were the 2 kingdoms. That's the way it persisted for a really, really long time. It really wasn't until, I think less than a century ago, or maybe a little bit more than a century ago, that people realized funguses are plant-like, but they're so different in so many ways and structured so differently that they shouldn't be in the plant kingdom, and they got split off. And then there were all these microscopic things that people were discovering when they were looking at the world through microscopes. And microscopic organisms do not necessarily follow the rules real well. You have some very plant-like, animal-like simultaneously, animal-like, fungus-like simultaneously groups. And then you've got the microscopic life whose cells are very different from our cells, that don't have nuclei, they don't have a bunch of other things. And so they needed to split those off into different groups. First, the bacteria, which are significantly different cells than us, so homology, very, very different. And then eventually, the protists, the single cell with cells like ours, and then they could argue about whether they were plants or animals or funguses, because that was kind of it, is that people got tired of the arguments over it should be classified as an animal, it should be classified as a plant, it should be classified as a fungus. Here, go into your own room and have your own arguments. So when you pick up different books of biology, you will see kingdom systems that vary from book to book. You won't see the 2-kingdom system anymore unless you're reading about biological history. You won't see the 3-kingdom system. You will probably see in basic biology books, so that's what we're going to deal with, you're going to see a 6-kingdom system, because that's our kind of entry level setup. But when I taught zoology, I used the same book over and over and over, and every edition was a different kingdom system. Is that this guy actually was in the microscopic group, that was his specialty. And there were editions where the microscopic group had been itself split into, I think, 6 separate kingdoms. And then, that didn't play out too well, and it shrank back down to 1 or 2 kingdoms in the next edition, or something else split off. I mentioned earlier Peripheral, the sponges. The sponges are considered a separate kingdom in a lot of zoology books, because they don't seem to share an ancestor with the animals, but it depends on who you ask whether they do or they don't. So they're in the animal kingdom in the introductory system, but there are 9 kingdom systems, 8 kingdom systems, 7 kingdom systems, 12 kingdom systems, 18 kingdom systems. Again, the names of the groups are still the names of the groups. The levels may be different, but the names tell you what you're dealing with. I first started teaching, it was a 5 system, a 5-kingdom system. And right off the bat, you'll see that it starts off confusingly. This is the level above kingdoms. Here's one kingdom split into 2 bigger groups. Now, how does that make any sense? It turns out it doesn't make any sense. But this used to be the 5-kingdom system. These have now been split off into their own separate kingdoms. And these 3 domains are considered to be above the kingdom system this week. Is that there was a big paper released over the summer that is finding that genetically, and that's the homology we're working with now. We're comparing sequences of DNA to look for similar structures. And they're thinking that these are actually ancestrally connected to these, that they're not a separate branch, they're a shared branch. So the domains may actually at some point go away. Who knows? Keep in mind, it's just labeling. It doesn't matter to the living things any more than it matters to whether we think viruses are alive or not. But I'm going to ignore the domains and use just your basic 6-kingdom system. First off, you need to know the difference between 2 basic types of cells. Prokaryotic cells, which are basically bacteria, the general name for them, and eukaryotic cells, which are the cells that we have. Prokaryotic cells are small. In some ways, they're simpler. There's more of them in the world than there are of any of the other guys, but they're a simpler system. And there's 2 kingdoms that are prokaryotes. Or there would be if I could spell prokaryotes. I'm going to give you a little bit more detail than I would a non-majors group. And even then, I'm lying to you to some extent, because it's too early to throw the complexity at you that I would need to in order to differentiate some of these groups. But as a general idea, prokaryotes are small cells. They don't have chambers inside them that are separate membranes from the outside membrane. What does that mean? Well, you can find prokaryotes with chambers, but the chambers are in foldings of the outside membrane. It's all connected. Whereas in our cells, you find a nucleus as a separate membrane enclosed chamber. You can find mitochondria, you can find all sorts of things in our eukaryotic cells. Now there are 2 kingdoms of prokaryotes, and here I really got to start lying. You've got the Monera and the Archaea. In the room with me right now, the vast majority of bacteria are Monera, is that they exist in the big wide world. There's way more species of them that you're going to run into. Archaea are less common in the wide world. You're going to find them in what are generally considered extreme environments. It's not a great differentiation. The real differentiation comes down to their biochemistry. Has to do with how they process DNA, how their cell walls are made, and I don't want to get into that this early in an introductory biology course. So this is the closest I can get to a separation between the 2 groups. But you find these guys in hydrothermal vents, you find them in hot springs, you find them in hypersaline lakes, you find them 8 miles down in the bedrock. you know, all sorts of really extreme environments that are very unfriendly to life. If you're gonna find life there, it's very likely to be archaea. And these are considered to be related to the very earliest of life, which is part of where the name comes from. But, you know, yeah, we got a few of them living in us, but most of the bacteria that live on and in us would be Moneras. Now, all of the other groups, the other 4 kingdoms, are eukaryotes. These are bigger cells. Some of them are single cells, but they are capable of being multi-celled systems. You don't find multi-celled systems that are made from prokaryotes. You find clumps. Very simple colonies in some cases, but not multi-celled systems. Eukaryotes, even though there are single-celled eukaryotes, if you're looking at something multi-celled, it by definition is going to be eukaryotic. So, you know, it's basically this stuff just opposite. And the rest of the groups are eukaryote groups. One of them is a single-celled group, mostly. You've got the Protista. And these are single-celled eukaryotes. Now, there was some sort of, I don't know, something happened. And the algae that largely grows in the open ocean, that are, if you look at them under the microscope, they are multi-celled, they're sheets of cells, but they're very, very simple sheets. They used to be considered part of the plants. At some point, the plant people kicked them out. I was not in on that discussion. I'm not a plant person. I don't really know. I just saw it. It just noticed that the books changed. And they were kind of exiled to the Protista from the plant kingdom. And the plant kingdom is now essentially, ancestrally, the land plants. The algae that are in the plant kingdom are descendants of land plants. You gotta be careful with photosynthetic. Plants are photosynthetic. And they're the only group that is all photosynthetic. But every group above us, above this, has photosynthetic species in it. Some of them have a lot of photosynthetic species in it. So there's photosynthetic Monera, and photosynthetic Archaea, and photosynthetic Protists. There's only one group, other than these weird algae, that's multi-celled and photosynthetic. And as I said, these are typically called the land plants. Now it gets weird. Animals, right? That's simple, they move. Well, that would be really good if all animals moved, but there are a whole bunch of different animals that are what are called sessile, that do not move as a typical part of their lifetime. And so move isn't that great a defining feature of the animals. The weird thing is that you need to actually separate these guys out from the funguses, because animals and funguses based on homology. I'm trying to make an adjective out of it and I can't get it. They're similar in a lot of ways, is that we and the funguses absorb and digest food. They do it across their outside surfaces, we do it from an internal chamber. So when I write the funguses here, I don't have room enough to put that, but it's just basically paralleling that. So you should write that down. So funguses digest and absorb across their outer surfaces. They secrete enzymes and then pull the nutrients in. Animals usually move. You can get away with saying that. It's not a characteristic of the whole group. And the same thing with funguses. Funguses usually are made up of a network of fibers called hyphae. Sometimes they pull a bunch of them together, like they do with mushrooms. But the actual fungus that the mushroom is actually a reproductive structure of is distributed through the soil as these single strands of cells, or strands of cells in a single row, that are called hyphae. And that's your typical formation of a typical fungus. And again, can't depend on it across the entire kingdom, so I gotta put the usually in. Same with usually move for the animals. Okay, I gotta squeeze a couple more things in. I know this is running long. There's been a decent differentiation between how you set up the branches. A lot of you have seen, and I should have an overhead for this, but I don't. I've got a link in the book to it. Your classic branching set of relatives that, where you got the ancestral groups going up to the descendant groups. And the closer branch is, the more related things are. That's a systematics approach. You connect the dots, connect the branches, and at every connection point, that's considered to be a common ancestral group that's split and then differentiated into the two groups farther up that may be split again. So that's systematics. That's the way they organize the trees. Then cladistics. They organize their trees kind of based on analogous. Well, it's homologous and analogous. Is that they do splits based on what they consider to be major evolutionary developments. So you got everything coming up this line has a vertebral column, but you split off here to the reptiles. You split off here to the mammals. Hair and mammary glands makes this root branch different. Then you've got well, it splits off for gestation. These are egg-laying mammals. I have to step back to get a better look at what I'm looking at. Is it gestation, where the young don't develop in eggs, they develop internally. Now, marsupials, they don't develop that long before they climb out and climb into the pouch and develop from there. And that's a split. And then you've got long gestation that you get in our group. But you can see where it's all connected to features. It's connected to characteristic features that does the splits. Is it the old, you'll see cladograms too that come in and there's a lot of right angles and connections and branches that way. That actually dates back to when this first developed as a way of looking at things. People loved to get the computers to draw these, but computers at that point were horrible at drawing diagonal lines. The printers couldn't handle it. So there was a lot of straight lines. And I actually do think that influenced how people looked at the splits, that the splits looked very much sudden rather than gradual evolutionary divergence. And so sometimes I think cladistics kind of falls into the trap of thinking that way. There's a link in the book to this called phyllocode. It's the new version of cladistics. And I will tell you, I periodically go back to the phyllocode site and go, okay, maybe somebody has explained it in a way that makes sense. Maybe somebody's put the basics in. And it never makes any sense to me, is that it doesn't seem to have caught on as a new organizational system. But there are some people that are very, very much into phyllocode, and you may wind up in a situation where that's true. I will mention that viruses do follow the basic hierarchical system, except that they do not go up above orders, I don't believe. That they have orders and families and genera and species, but they don't have classes and, I don't know, it's just the weirdness of the way the world works. Okay, that is it for chapter 2.